Hello everyone. Last time we talked about the overactivity immune response, autoimmune disorder. Today we are going to talk about the immunodeficiency, which are manifest clinically by increased susceptibility to infection, as well to certain forms of cancer. Immunodeficiency could be classified into immunodeficiency disorder, which are genetically determined, and secondary immunodeficiency, which may arise as a complication of cancer and infections. As a group, the secondary immunodeficiency are more common than the disorder of primary genetic origin. Thus, we will discuss the most important secondary immunodeficiency disorders and go through a more detailed description of acquired immunodeficiency syndrome, AIDS. AIDS is a disease caused by human immunodeficiency virus and is characterized by profound immunosuppression that is to opportunistic infection and secondary cancers. AIDS was first recognized as a distinct entity as recent as the 1980s. An estimated 7.7 million people have become infected with HIV since the start of the epidemic. In 2019, globally, 38 million people was living with HIV, and around 96,000 people died from AIDS-related illness worldwide. The three major routes of HIV transmission are sexual contact, parental inoculation, and the passage of the virus from infected mother to their newborns. Sexual transmission is the dominant mode of the infection worldwide, account for more than 75% of all HIV cases, while mother-to-infant transmission is the major cause of pediatric AIDS. The outer shell of the virus is called the envelope containing spikes of the glycoprotein GP120 and GP41, which allowed HIV to lock onto the CD4 receptor and co-receptor CCR5 or CXCR4 on host cells. Immune system cells include T lymphocytes, monocytes, macrophage, and dendritic cells, each has a CD4 receptor on its surface. HIV infects cells by using the CD4 molecules as a receptor to multiply and spread throughout the body. This process, which is carried out in 7 steps called the HIV life cycle, includes binding, fusion, reverse transcription, integration, replication, assembly, and budding. Therefore, the life cycle of HIV consists of infection of host cells, integration of the pre-virus into the host cell genome, activation of viral replication, production, and the release of the infection virus. Completion of the viral life cycle in latently infected cells occurs only after cell activation, and in the case of the most CD4 T cells, activation leads to death of the infected cells. This results in infection and subsequent loss of CD4 T cells, as well as impaired function of surviving helper T and other immune cells, including macrophage and dendritic cells. Mechanism of immunodeficiency: loss of CD4 T cells, T cells death during viral replication and budding. Apoptosis occurring as a result of chronic stimulation decreased seismic output. Functional defects. Defective macrophage and dendritic cell functions. Destruction of the architecture of lymph node tissues. Morphological biopsy specimen from enlarged lymph nodes in the earlier stage of HIV infection reveal marked follicular hyperplasia with disease progression. The frenzy of B-cell proliferation, suicides, and give way to a pattern of severe lymph node involution. The lymph nodes are depleted of lymphocytes, and the organized network of follicular dendritic cells is disrupted. The germinal centers may even become hyalinized. Finally, those bottom out lymph nodes are atrophic and small. CD4 T cells, macrophage, and dendritic cells play a major role in protecting the body from infection. Over time, these cells are killed by HIV, and the body's ability to recognize and find some type of infection begins to decline. If HIV is not treated, the loss of those cells leads to the development of serious infection caused by organisms, which will not normally lead to infection in healthy individuals. This opportunistic infection accounts for the majority of deaths in untreated AIDS patients. For example, tuberculosis remains the leading cause of death among the people living with HIV, comprising around 1 in 3 AIDS-related deaths. It's estimated that 44% of people living with HIV and TB are unaware of their co-infection and are therefore not receiving care. Furthermore, patients with AIDS has a high incidence of certain cancers: sarcoma, B-cell lymphoma, cervical cancer in women, anal cancer in men. It's estimated that 25 to 40% of untreated HIV-infected individuals will eventually develop a malignancy. Many of these tumors are caused by oncogenic DNA virus, including sarcoma herpesvirus, EBV, and human papilloma virus. For example, in HIV-infected individuals, the sarcoma is usually widespread, affecting the skin. Morphological sarcoma is characterized by a proliferation of spindle-shaped cells that express markers of both endothelial cells and smooth muscle cells. To summarize, as a typical acquired immunodeficiency disorder, AIDS is caused by HIV infection. That cystic infection and malignant tumors by loss of CD4 T cells, macrophage, and dendritic cells. Let's move on to the next topic, graft rejection. This is a process in which T lymphocytes and antibodies product against graft antigens with and destroy the grafts. Graft rejection is classified into hypoacute, acute, and chronic on the basis of clinical and pathological features. First, hypoacute rejection occurs within minutes or hours after transplantation, resulting in rapid graft loss. Hyperacute rejection is mediated by performed antibodies specific for antigen on graft endothelial cells. Therefore, all arterioles and arteries exhibit acute fibrinoid necrosis of their walls and narrowing or complete occlusion of their lumen by thrombi. Second, acute rejection may occur anytime from the first weeks to 3 months after the transplant. It also may appear suddenly months or even years later, after immunosuppression is tapered or terminated. Acute rejection is mediated by T cell and antibody that are activated by alloantigen in the graft. Depending on whether it is the T cell or antibodies, acute rejection is further classified as acute cellular rejection or acute humoral rejection. Morphologically, in acute cellular rejection, lymphocytes infiltrate the interstitial and perivascular compartment of the allograft, resulting in graft cell damage and necrosis. CD4 T cells may direct destroyed graft cells, or CD4 T cells secrete cytokines and induce inflammation, damaging the graft. T cells may also react against graft vessel, leading to vascular damage. In acute humoral rejection, antibodies bind to vascular endothelium and activate complement via the classic pathway. The resultant inflammation and endothelium damage cause graft failure. Finally, chronic rejection is an indolent form of graft injury that occurs over months or years, leading to progressive loss of graft function. Chronic rejection manifests an interstitial fibrosis and gradual narrowing of graft blood vessel. In both situations, the culprits are believed to be T cells that react against graft alloantigen and secrete cytokines, which stimulate the proliferation and activation of fibroblasts and vascular smooth muscle cells in the graft. To summarize, hyperacute rejection is mediated by performed antibody specific for antigen on graft endothelial cells. Acute cellular rejection is caused by T cell destroyed graft parenchyma by cytotoxicity and inflammatory reaction. Acute humoral rejection is manifested mainly by damage to small blood vessels. Chronic rejection is dominated by vascular changes, often with internal thickening and vascular occlusion. They are the reference, and thank you for your attention.